Hi all, I am Sahu Anjana, pursuing MSc Microbiology in Central University of Tamil Nadu. Today I am going to discuss on microbial degradation, focusing mainly on two components, petroleum hydrocarbons and xenobiotics. Coming to introduction, petroleum hydrocarbons and xenobiotics are one among the major components that are polluting our environment by several human activities such as oil extraction, misuse of chemicals which are used in agriculture and industrial waste, and these components are accumulating in our environment and eventually leading to the pollution. Why we are considering them as pollutants? Because they are very hard to break down and remain in the environment for a longer period of time, which are eventually leading to threat to the biodiversity, water quality, soil health and human beings as well. Microbial degradation is a bioremediation method where these microorganisms break down these harmful pollutants into very less toxic substances, offering an eco-friendly way to clean up the contaminated areas. Petroleum hydrocarbons are organic compounds consisting of carbon and hydrogen, originating from crude oil and natural gases. They include fuels like gasoline and diesel, which can contaminate soil, water, and air, harming the environmental quality. So, the type of petroleum hydrocarbons involves alkanes, which are very straight or branch chains, easier for microbes to degrade. And the next is aromatics, which are ring-shaped structures and very toxic in nature and also more resistant to degradation. And the next is resins, which are very complex heavy molecules, highly resistant and difficult for microorganisms to break down. So here you can see an image where it depicts the fractional distillation of crude oil is heated in a column to separate it into products like gasoline, diesel and lubricating oil based on their boiling points. After separation, each fraction has specific uses. Some of them are like gasoline is used for as fuel for cars, diesels, and diesel are powers the trucks and machinery, and kerosene is used in jet fuel and heating, lubricating oils to reduce the friction in engines. What is the microbial role in degradation process? Bacteria such as Pseudomonas rhodococcus can metabolize hydrocarbons by taking them as carbon source, which involves two main pathways based on oxygen presence, aerobic and anaerobic pathways. In aerobic degradation, oxygen-dependent enzymes like alkane hydroxylases and dioxygenases initiate the breakdown process. In anaerobic degradation, which is in the absence of oxygen, is often very slower as it involves different microbial pathways and is crucial for oxygen-limited environments. Let's see the factors that affect the degradation process, such as environmental factors, which involves temperature, pH, oxygen levels, and nutrient availability, which can influence the degradation process. And we can also enhance the degradation process by biostimulation, which involves addition of nutrients to the enhanced microbial activity, and bioaugmentation, where specialized microorganisms are introduced to speed up the degradation process. Let's discuss some of the microorganisms which can enable to degrade the petroleum hydrocarbons in the environment. First one is Pseudomonas aeruginosa, which is a gram-negative bacterium found in soil and aquatic environment which are contaminated with oil. It is able to degrade complex hydromolecules through the production of biosurfactants, which is going to emulsify the oil. The optimal temperature range for its biodegradation activity ranges from 30 to 37 degrees. Pseudomonas aeruginosa uses oxygenase enzymes that introduce oxygen into hydrocarbon molecules, breaking them down into simpler molecules. And by this mechanism, Pseudomonas can able to degrade the petroleum hydrocarbons in the environment. In the next microorganisms helpful in the biodegradation of petroleum hydrocarbons is Rhodococcus species, which are bacterium found in oil contaminated soils, marine and terrestrial ecosystem. It can able to degrade wide range of hydrocarbon compounds including alkanes, aromatics and complex petroleum substances. Rhodococcus species utilizes cytochrome P450 monooxygenase enzymes to oxidize the molecular structure of these hydrocarbons. These oxidation steps break down the large recalcitrant hydrocarbon molecules into smaller, more biodegradable intermediates. and that optimal temperature range for its growth and hydrocarbon degradation activity is 25 degrees to 35 degrees centigrade. This microbial process results in the metabolism of contaminants into more biodegradable intermediate compounds, contributing to the effective bioremediation of oil polluted environments. And the next microorganisms is Alkanivorax brocumensis, which is a gram-negative rod-shaped bacterium known for its ability to degrade hydrocarbons. Its primary mechanisms involve specialized metabolic pathways and unique membrane-associated alkane hydroxylation system that allows it to efficiently break down linear and branched alkanes. This bacterium is commonly found in marine and coastal environments contaminated with oil. It thrives in the temperature range of 20 to 30 degrees centigrade, which provides the optimal condition for its hydrocarbon degrading capabilities. As Alkanivorax brocumensis metabolizes the alkane compounds of the oil pollutant, it plays a role in a bioremediation of contaminated marine systems. Its specialized hydrocarbon degradation pathways makes it a highly effective microorganisms for cleaning up oil spills and restoring the health of affected aquatic environments. And the next microorganisms is Aspergillus niger, which is a fungus that can thrive in contaminated soil environments. Its primary mechanism for degrading hydrocarbons involves the production of extracellular enzymes, particularly lignolytic enzymes. These non-specific oxidative enzymes allows Aspergillus niger to break down complex aromatic hydrocarbons compounds. This fungus is able to grow and utilize these enzymes most effectively in a temperature range of 35 to 45 degrees centigrade. Through this enzymatic degradation process, Aspergillus niger is able to metabolize and remove a variety of aromatic hydrocarbon pollutants from contaminated soil ecosystem. Its versatile nature mechanism makes it a useful microorganism for bioremediation efforts targeting complex oil-derived contaminants. And the next environmental pollutant is xenobiotics. These are the compounds that are foreign to an organism or ecosystem and not naturally produced or expected to be present in the environment. They include wide variety of substances like the drugs ended up in wastewater from pharmaceuticals, agricultural chemicals from pesticides, and solvents, metals from chemical industries, petroleum products which are environmental pollutant, personal care products things like soaps and cosmetics, food additives such as synthetic preservatives and other contaminants. And these xenobiotic compounds can sometimes be very harmful to the environment and living organisms due to their toxicity and also persistent nature in the environment. Understanding the different types of xenobiotics is very important to access the environmental impacts and also to develop some strategies for their remediation. Now let's see the microorganisms which are helpful in the degradation of xenobiotic compounds. The first one is a fungus named as Phanerochaete chrysosporium, which is commonly found in forest, soil and environment involved in wood decomposition. Its primary mechanisms are for breaking down the xenobiotics compounds involves majorly two key enzymes, lignin peroxidases and manganese peroxidases. These extracellular non-specific oxidases enzymes allow the chrysosporium to degrade a wide range of recalcitrant organic compounds including those that can accumulate through the food chain. These fungus thrive in the environment optimal temperature range of 37 to 45 degrees centigrade, which enables its enzymatic degradation capabilities. And by producing these enzymes, these fungus can eliminate the xenobiotic compounds present in the environment. The next microorganisms here we are discussing about is Pseudomonas putida, which is a gram-negative bacterium that has the distinction of being the first organisms to be patented for its bioremediation capabilities. One of its key ability is by degrading the organic solvents like toluene. The mechanism behind this lies in Pseudomonas putida is plasmid-mediated degradation pathway. These specialized genetic elements allow the bacterium to produce the necessary enzyme systems to completely mineralize the complex synthetic compounds. Pseudomonas putida is commonly found in contaminated industrial soil environments, and it thrives in the optimal temperature range of 25 to 30 degrees centigrade, which supports its efficient xenobiotic degradation capabilities. And the next microorganisms here we are discussing is Rhodococcus rhodocrus, which is a gram-positive bacterium that utilizes specialized cytochrome P450 monooxygenase enzyme system to degrade a variety of xenobiotic compounds. This heme-dependent enzyme plays a crucial role in detoxification and bioactivation of complex synthetic molecules. This bacterium is commonly found in polluted industrial waste sites. It thrives in the optimal temperature range of 28 to 35 degrees centigrade, which supports the efficient xenobiotic degradation capability. Here are some common degradation methods for both petroleum hydrocarbons and xenobiotics. The first one is oxidation, where the microorganisms utilize some oxidative enzymes such as monooxygenases and dioxygenases to introduce oxygen atoms inside the hydrocarbons. This will facilitate the increase in the polarity of the molecules and also helpful in the degradation of the hydrocarbons. And the next is hydroxylation, where the addition of hydroxyl group is taking place and which will create more polar compounds which will helpful in the enzymatic degradation. And also this enables the transformation of hydrophobic hydrocarbons into hydrophilic forms, which is a very crucial step in the metabolic pathways for both petroleum and also xenobiotic degradation. And the next process is conjugation, where the microorganisms attach specific molecular groups to the pollutant molecules. Some common conjugation process includes glutathione conjugation. So basically this process involves in increasing molecular weight and polarity, facilitating excretion and reducing toxicity. So by this, transformation of complex molecules into more manageable metabolites intermediates will be formed. And the next is cleavage of chemical bonds. Microbial produce some enzymes which will strategically break specific chemical bonds present in the hydrocarbon and also xenobiotic molecules. So this break large complex molecules into very smaller molecules so that it will helpful in the degradation by enzymatic mechanism from like targeting carbon-carbon and carbon-hydrogen bonds. And the last method is transformation of molecular structures. Microbes systematically modify their molecular structures through enzymatic process. It involves rearranging atomic configuration to create more biodegradable intermediates, transforming ring structures to aliphatic chains and complex organic molecules, which enables complete mineralization of these complex or synthetic molecules. So these are the mechanisms which are working in a synergical manner, allowing microorganisms to progressively break down and metabolize complex petroleum hydrocarbons and xenobiotics, ultimately converting them into simpler, less harmful compounds like CO2, water, and biomass. Let's see how fuel invention led to the discovery of hydrocarbon degradation microorganisms. In 1900s, Rudolf Diesel demonstrated an engine powered by peanut oil, marking a significant milestone in fuel technology. However, an unexpected discovery soon followed. Microorganisms were found to interact with the fuel in a surprising ways. One of the key bacterial strains involved in this process was Bacillus thermophilus, which is discovered by Dr. Martinus Bijanek in industrial storage tanks. This discovery marked the first systematic documentation of microbial hydrocarbon degradation. The bacteria were capable of metabolizing the complex hydrocarbon compounds present in this oil, transforming what was once considered as contamination into valuable scientific breakthrough, showcasing the potential of microorganisms in environmental cleanup and fuel technology. And here in the conclusion part, it's highlighting that microbial degradation offers a sustainable approach to tackling petroleum hydrocarbons and xenobiotic pollution. And bacteria and fungi transform harmful pollutants into very harmless substances. By optimizing environmental conditions, biodegradation rates can be increased. And genetic engineering has the potential to enhance microbial efficiency. Bioremediation serves as an eco-friendly method for managing pollution and safeguarding both ecosystem and human health. These are the some references, and I would like to thank Kavya ma'am for giving me this opportunity, and thank you all.